Good. I'm just going to launch here. Thanks. Thanks for having me, and really, really apologize for not being there in person. Rob, Richard, would have been good to see you face to face. When I saw the CTC meeting going back to Memphis, I got nostalgic, and I wanted to tell us all about the history of EQTL and molecular trait mapping, but it takes more than 15 minutes. So I want to remind everybody, and for those of you who are as older than Richard, you'll remember this paper from 2001 by Jansen and Knapp, where they proposed that doing molecular profiling in genetically segregating populations could be really cool. And I think this session today proves that they were right. And I just wanted to point out that in 2001, we were still doing back crosses. We were using RFLPs on gels for markers, and the best we could do for gene expression was a microarray. So let's go ahead and fast forward 23 years. I would like to tell you today about some work that I did with Tian Zhang from the Gigi lab and the whole crew, including Fernando and Marty from UNC, to look at a multi-omics analysis in genetically diverse mice that identifies drivers of protein phosphorylation. And you notice this little subtext here. That's because I wanted the paper to be published, but the genetically diverse mice are the most critical piece of the equation. Many of you know these mice. I won't show off and name them all, but I will point out the giant fat one in the background. That's NZO. And this is not really WSB. This is fake WSB, because real WSB doesn't sit still for a picture. So what we did is some proteomics, but we didn't do them on the 8 founders. We took Amelie. This experiment could be done as well in outbred or inbred mice, but I'll show you where the inbred comes in really handy. We took inbred collaborative cross strains. We actually obtained the tissues from UNC on 58 strains. For each of these strains, we had 1 male and 1 female animal. And I think really the advantage of inbred strains in this context is that we could make a paired direct comparison between male and female, really improved our power to detect sex differences. So 58 CC strains, 1 male and 1 female from each. We collected heart, kidney, and liver, and we carried out proteomics and phosphoproteomics. Proteomics, I'm sure you're all familiar with. We heard some talks about it this morning. We use TMT mass spec technology to quantify peptides. The individual peptide measurements are rolled up to whole proteins. We can also detect individual peptides by their shift in molecular weight that have been phosphorylated. So we have quantified phosphorylated peptides. These peptides are components of proteins. And because if a protein abundance increases, the peptide abundance of all the peptides in that protein should increase. We actually calculated something we call an adjusted phosphopeptide abundance, which is simply the regression of the phosphopeptide on the abundance of the protein, but we modified the protein abundance by pulling out all the peptides that we thought could be phosphorylated. And that's why, we detect about 7,000 proteins, about 5,000 phosphopeptides, and we lost a few in the adjustment phase, but we're still able to look at thousands of phosphorylated peptides. And what we're going to do is we're going to use some genetical genomics to learn what we can learn about them. So I think it's easiest to illustrate what we're trying to do here by considering the sex differences in protein phosphorylation. You can imagine that there could be phosphorylation sites in the genome on proteins that are sex-specific for whatever reason. We know that proteins have sex-specific expression, and if you calculate this is the standardized sex effect, and I forget which way it goes. Negative must be higher in male, positive higher in female. If you look at these tissues, heart has relatively low sex specificity. Kidney has really high sex specificity, no surprise, and the liver is just right. So we've got a bunch of, different levels of sex specificity in proteins. If we go down to the phosphopeptide level, not so much. There's still quite a lot of sex specificity, though, even though, I mean, the kidney numbers are ridiculous, but there's still a lot of sex specificity in the phosphopeptides. And all I wanted to use this for is to illustrate a point. We looked at lots of proteins. Here's an example, a protein called LDHD that has higher expression in males. We looked at a particular phosphopeptide. This is the serine at position 23 that's phosphorylated. And we see that the phosphorylated peptide itself has higher abundance in males. But after we do the regression adjustment of accounting for the level of the protein, we end up with an adjusted phosphopeptide that has no significant difference. And what we conclude is that sex has a direct effect on protein abundance, and protein abundance has a direct effect on the peptide abundance. Makes a lot of sense. So if we adjust for or account for the protein abundance, it blocks this direct connection between sex and the peptide. We saw hundreds of examples, though, of cases like this, where in this CGREF1 protein, slightly higher expressed in the males, but the phosphopeptide at serine 272 is much more lowly expressed in the males. And after adjustment, things don't change. And what we conclude here is that sex may affect the protein. In fact, it does in the opposite direction, but really the strongest effect here is a direct effect of sex on the phosphorylated peptide. And what we assume is that somehow, in a way that we aren't claiming to understand yet, the sex of the individual affects the level of phosphorylation of this particular peptide in this particular protein. So that's all well and good. But what we really wanted to do today was talk about genetic regulation of phosphorylation. Took a quick look at the heritability of these things. Proteins, highly heritable. Peptides, not so much. Adjusted phosphopeptides, even less. But still, there are hundreds of phosphopeptides that are genetically heritable. So we did the pQTL mapping. These are very familiar plots for you. We've seen several in this session. This is the position of the pQTL, the genetic association, and this is the position of the protein itself in the genome. We see the prominent cis band, and we see some off diagonals. So these are typical pQTL maps. We can also map peptide QTLs, and the colored dots here are the peptide QTLs, and the dark black dots are QTLs for adjusted phosphopeptides. So you can see in the peptides, we have still this nice diagonal, phosphopeptides, this nice diagonal line. In the adjusted phosphopeptides, there are really relatively few cis, and many of them are distal. And there are trans bands for the phospho-QTLs. Here's one on chromosome 6. I'm going to point to this particular trans band, which doesn't look that exciting here, but there is a really important trans band here on chromosome 2 that's present in all of our tissues, as I'll show you momentarily. So this is our model, that we have a genetic variation, which we call a QTL. The QTL could affect the abundance of a protein, and the protein abundance is going to directly affect the abundance of the phosphopeptide. But the QTL could also reach out and affect other factors, including obvious things like kinases, phosphatases, and other regulatory factors that might influence the level of phosphorylation and have an indirect effect on the phosphopeptide. So we can use genetic mediation analysis to identify the mediators, the intermediate steps between the QTL and the phosphopeptide. And the first example is the boring one, and that is the local phospho-QTL are largely just mediated by the abundance of their own protein. This is an example of a protein called Gas2. It has a really strong local QTL. And if we model the QTL effect on the protein and the protein effect on the phosphorylated peptide at serine 283, we see that the proportion of variance explained in the protein by the QTL is 80%, and the proportion of variation in the peptide explained by the protein is 97%. There's not much doubt that this is a direct path. There's no variation left over to be explained. And you can see this is a low PWK effect. I know you all have these colors memorized like I do, low PWK effect. And in fact, the protein abundance is low and the peptide abundance is low. And there are hundreds of examples like these, including TPMT and MTX3. The interesting ones are the distant phospho-QTL. These are largely not mediated by protein abundance. And here's an example of a protein, MCAT, which is physically located on chromosome 15. But when we mapped the phospho-QTL for serine in position 41, we find that it maps to chromosome 2. The allele effects of the distal QTL on chromosome 2 are low NZO. Remember the big fat mouse hiding behind the other mice? Low expression in NZO. And then we did a little trick that I don't have time to explain today, but we scanned through all of the proteins and we asked which of the proteins that we detected could potentially be mediators of the phosphorylation effect at MCAT. And there was one that dropped out of the bunch pretty clearly. It's called PDK1. This is pyruvate dehydrogenase kinase 1. Okay, we'll come back to that. And what we found is that this QTL on chromosome 2 doesn't really have an effect on the protein abundance, or the protein doesn't have much an effect on the phosphopeptide, but the QTL has a huge effect on PDK1 abundance. It's actually a local QTL for PDK1, and PDK1 in turn, kinase, has a big effect on the phosphorylated MCAT. This is the PDK1 expression, protein abundance, and we can see the abundance of PDK1 is low, associated with NZO alleles, and the adjusted phosphopeptide is also low. PDK1 is on chromosome 2. It's one of the hotspots I pointed to. And in fact, at high stringency, and I forget how high right now, but we did high stringency on these maps at first, and we saw about 7 other loci hitting multiple tissues that all mapped back as being mediated by PDK1. So there are a lot of phosphopeptides whose expression level is not mediated by their protein abundance, but it is mediated by this kinase. Okay. There are more complex examples of phospho-QTL regulation, and it shouldn't be surprising to learn that there are examples where we have paired distal and local effects, and the local effects generally go through the protein itself and have a direct effect on the peptide, and the distal usually go through some other molecule, some other molecular intermediate, the mediator or the phosphorylation. And in this case, this CDC 14B is colored green because we didn't actually detect the protein. We only detected the RNA, but I wanted to make the point that you can do this trick with RNA as well as protein. Protein's better. It's better. Good. Now I'm at the punchline. So let's go back to this PDK1. This is a really important little kinase. This pyruvate dehydrogenase kinase 1 activates or deactivates pyruvate dehydrogenase. And this is the gatekeeper in the mitochondria that says pyruvate should stay in the cytosol and go into biosynthesis, or pyruvate should come into the mitochondria where we can burn it for energy. So if you're a big fat mouse, maybe you're not regulating your mitochondrial metabolism appropriately. And I just wanted to show you that when we lowered the stringency and we asked, what are all the phosphorylated peptides that are potentially regulated by PDK1? This is the list. the list overlaps quite a bit between the tissues, and I think where it doesn't overlap, we just fail to detect them. One of the flaws of this study is that we did not go deep enough in the phosphopeptide characterization, but we did capture some interesting results like pyruvate dehydrogenase subunit A1. That's a ringer. We don't need to validate that. It's true. But all of these other phosphorylated peptides, a few of them are known, but most of them are novel. And all of these proteins localize to the mitochondria where they need to be to see this kinase. So I'm really excited that we've been able to use genetical genomics to identify targets, downstream targets of PDK1 that were previously unreported. We looked at some other large mediators of lots of phosphorylation events. This is a kinase, yep. And this is another example of lots of regulation. And I think I'm being told to wrap it up. So let me massively thank Greg Keel and Tian Zhang, who really were the hardworking people here. You can find our data. This is our paper, and I thank you much.